One of the most powerful and useful features of ASAP is the ability to combine accurate tissue optics with realistic biogeometry characterizations. Let's look at a few such examples. This bladder model that you see was created by the Brody School of Medicine at East Carolina University from CT scans of an actual patient. These CT scans were then reconstructed into a three-dimensional model, which we can import into ASAP, apply optical properties to, and trace rays through in order to get an idea of the light distribution within the wall of the bladder. In order to more adequately visualize this, let's take a look at this in 3D. We can remove portions of the bladder in order to examine the scatter and absorption within the wall of the bladder. As you can see, we have an isotropic point source that is propagating through a saline solution inside the bladder, and scattering is occurring within the wall of the bladder. And we can visualize this using our voxel system. We can actually see absorption in the medial, sagittal, and transverse planes. We can visualize this as well as a single slice through the center of the bladder. This is important for applications such as photodynamic therapy, where we would be creating a probe that was placed in the bladder and illuminating the interior of the bladder in order to activate a photosensitizer. In a lot of these applications in the past, there have been problems with creation of fibrosis or scarring in the inside of the bladder, basically turning a non-invasive procedure into an invasive procedure later on when they have to remove the scarring. What we can do is we can examine the light distribution on the inside of the bladder and compare that to previous simulations where only a spherical bladder was used as an approximation, and determine, based on the structure of the bladder, what the ideal probe shape is in order to get uniform illumination on the interior of the bladder. Now let's take a look at a few other examples of how geometry has influenced design in biooptics using ASAP. This rat brain model, which was created based off of an MRI slice of the rat brain, was used by Stanford to characterize a system to examine activation of specific regions of the brain by examining blood oxygenation concentrations so that we can create a non-invasive method of examining the activation of specific areas of the brain without having to use devices such as MRI. This chest wall example was used by the Brody School of Medicine to examine photodynamic therapy treatment in a post-mastectomy patient. They've used an external LED-based source and are illuminating the chest wall in order to activate a photosensitizer and remove any potential cancerous cells that are left over after the mastectomy. Complex biological structures are not the only structures that may be interesting in biooptics. For instance, this complex tracheal mesh is used in patients that have tracheal or esophageal cancers in order to hold the esophagus or trachea open and facilitate breathing or swallowing. Now, this mesh can become intertwined with the tissue of the trachea or esophagus and requires photodynamic therapy treatment in order to remove excess tissue and reopen the passage. As you can see, this is an incredibly complex mesh design, and therefore it's important to be able to model each individual portion of this mesh to get an accurate depiction of the optics that are necessary in order to effectively treat this situation. Finally, we can see here a finger model with realistic optical properties for stratum corneum, epidermis, dermis, musculature, and bone. And this model is integrated with a blood oxygenation sensor, including a parabolic mirror, an LED, and underneath a detector, so that we can examine the level of blood oxygenation within the dermal layer of this particular model. This is a great example of how we can create a complete virtual prototype using both a biologically relevant structure as well as realistic optics. And these are just a few examples of how tissue models can be applied to complex biological structures in ASAP to help us better understand complex optical systems for biooptics.